Hello and welcome to Nikolai's genetics lessons, and today is going to be my second video about how to read Manhattan plot and how to understand it, all these graphs, dots and signals. Before I will go into details, let me tell you something about the theory. As far as probably many of you know, that 99.9% of our genomes between people are identical, and only 0.1% is different. What something makes us different from each other? While 0.1% sounds tiny, our genome is so massive that this 0.1% accounts for roughly 3 million differences, mostly SNPs, between you and stranger. SNPs stand for single nucleotide polymorphisms. For example, imagine that we have a small fragment of the DNA and the sequence is going to be as follows. So G, C, T, A, and T. And in other person, the sequence is going to be in the same fragment, is going to be as follows. G, C, A, and T. So as you see, these two sequences are different in this single spot, which we call single nucleotide polymorphism. When scientists look specifically at such SNPs, they found that typical individual carries between 4 and 5 million such SNPs that differ from standard reference human genome. Most of these SNPs, about 90%, are common variations found in many populations, and a small percentage are unique to you or your immediate family. Now let's return to this graph, and as you see, at the very bottom, we see a solid color, but actually it consists of many dots here. We analyzed between 500,000 and 1 million such SNPs, but here they just become looking as solid because of the sheer number of these dots. Even though humans are 99.9% the same, those millions of tiny SNPs differences are what determines your height, your risk of certain diseases, and how muscles respond to training, and many other traits, including phenotypical traits like, for example, size of your nose or color of your eyes, and many other traits, including how you react to or respond to certain medicine. So a Manhattan plot is just a standard bioinformatic tool which is used in genome-wide association studies, and it helps visualize which parts of the DNA or specific genes or SNPs are linked to particular trait or disease. Now take a look at the horizontal x-axis. It represents the human chromosomes 1 through 22 autosomes, and each dot is genetic variation or SNP, and the different colors you see here simply help to distinguish one chromosome from the next, and sometimes you may see them just in two colors. Colors here doesn't matter, just to distinguish one chromosome from another. And on the y-axis or vertical, this is your familiar minus log 10 of p, and if you don't know what it is, I will make another video in which I'm going to explain how to calculate and the logic behind it. And the higher the dot, the smaller the p-value. And smaller p-value means higher statistical significance. Essentially, there is a lower chance that the connection is just a coincidence. Now let's talk about the lines which you see here, blue and red. For example, blue line usually represents the suggestive significance threshold. For example, P, in this case, P equals to 10 in the power negative 5, or 0.00001. And red line, usually set to 7.3. This is the gold standard in genetics in genome-wide significance, and anything above this line is considered real scientific discovery. And 7.3 in our calculations is roughly equal to 0.0000005. As you see, this number is much smaller than this number, which represents blue line. This number means that the chance of false positive is only 5 in 100 million, or 1 in 20 million, which is very, very small number, and we can say that this finding is going to be very significant and reliable. The most exciting parts are the peaks that hit the sky or past this red line. For example, take a look at chromosome number 6, which is shown here in the gray color, and you can see a massive gray skyscraper shooting past level 20. This is incredibly strong signal, and this area likely contains a key gene for the trait you are studying. You also can see that chromosome number 2 and number 5 also would be candidates, and they are confidently cross the significance threshold. You also can see labels here, so each single nucleotide polymorphism has its own label. When you see multiple dots which would line up like this usually means that this is going to be multiple single nucleotide polymorphism within single gene, and of course they are going all to be associated with a certain gene in which we can find them. And for example, if we take a look at chromosome number 10, we also see SNP here, which is barely above this threshold line, and that means that we still have to run more tests on this, if this is going to be strongly associated with our trait or not. But as for these two, we definitely can say that they have some association with a trait or whatever we're studying. Now let's move to this graph B. This is QQ plot. The small graph is quantile plot, and the pink straight line represent what we would expect if all results were random or noise, and the black dots, which curving sharply upward on the right side, confirms that you have a real biological signals in your data rather than just statistical errors. And I'm going to make a couple more videos on how to read Manhattan plot, how to analyze it. So keep watching, and this is all for today, and see you in the next video. Goodbye.